Bismillahir Rahmanir Rahim. Welcome again to Derma Immunology. I am Naglaa Elmongy, Professor of Dermatology, Cairo University. Our video today is the third video about skin fungi in health and disease, role of interleukin 17, and I'm going to focus in this video about type 17 immunity as an antifungal defense. T-cell responses against skin fungi typically show a type 17 profile consistent with the known role of interleukin 17 in defending against fungi and other extracellular microbes in barrier tissues. This was initially observed in T cells reactive to Candida albicans, which displayed a T helper 17 profile in the peripheral blood of healthy individuals primed by the commensal fungi. Also, T helper 17 cells reactive to Candida albicans are found in the skin. In healthy individuals, T helper 17 cells are present, targeting a wide range of commensal and environmental fungi. Among these, T helper 17 cells reactive to malassezia are especially abundant, reflecting the prevalence of this commensal yeast across the entire body surface. Also, experimental studies support the significant role of interleukin 17 cytokines in protecting against infections caused by Candida albicans, Candida auris, Malassezia species, and dermatophytes on the skin. Interleukin 17 cytokines operate through the interleukin 17 receptor signaling in epithelial cells and fibroblasts, enhancing the production of antimicrobial peptides, some of which are vital for fungal control in vivo. Also, interleukin 17 signaling plays a key role in preserving skin barrier integrity, promoting tissue repair and wound healing, all of which are essential for effectively managing fungal colonization on the skin. And even though interleukin 17 can increase the expression of chemokines that attract neutrophils, encouraging neutrophil response isn't the primary function of interleukin 17 during fungal commensalism. This is because during normal conditions, despite the ongoing activity of T helper 17 cells, neutrophils do not enter colonized tissues. The protective effects of interleukin 17 are at least in part shared with those of interleukin 22. T helper 17 cells which target skin fungi often produce both interleukin 17 and interleukin 22. And in a model of oral candidiasis, it was suggested that interleukin 17 receptor expression may be enhanced by interleukin 22. In addition to CD4 positive T cells, other lymphoid cells also contribute to the overall interleukin 17 response to commensal fungi, including CD8 positive T cells, dermal gamma delta T cells, and TCR negative innate lymphoid cells. And although these various interleukin 17 producing cell types respond to the same activating and polarizing cytokines, interleukin 1, interleukin 6, and interleukin 23, they show a distinct response kinetics. Innate lymphoid cells type 3 and gamma delta T cells tend to be activated more rapidly compared to CD4 positive and CD8 positive T cells. The production of interleukin 17 by human gamma delta T cells in response to commensal fungi has been observed, and in untreated HIV positive patients with Candida co-infections, interleukin 17 producing gamma delta T cells were suggested to partially compensate for the reduced CD4 positive T cells. Also, CD8 positive T cells generate interleukin 17 when exposed to skin fungi. The cytokines responsible for triggering type 17 immunity, typically interleukin 23, interleukin 6, and interleukin 1, are mainly produced by tissue-resident myeloid cells, including antigen-presenting cells during adaptive T cell priming, in response to direct sensing of the fungus by these cells. Additionally, other cell types especially epithelial cells, may also play a role in generating interleukin 17 inducing cytokines. This was demonstrated in an acute Candida albicans infection model using a highly virulent Candida albicans strains, which causes extensive cellular damage leading to the release of interleukin 1 from the injured cells. Also, the nervous system has been implicated in initiating type 17 immune responses against fungi in the skin, as observed in cases of cutaneous candidiasis. In the skin of experimentally infected mice, sensory neurons were observed to directly detect Candida albicans and trigger the generation of the sensory neuropeptide calcitonin gene-related peptide. Calcitonin gene-related peptide in turn induce CD301b positive dermal dendritic cells to produce interleukin 23, instructing gamma delta T cells to produce interleukin 17. Also, interestingly, the activation of TRPV1 positive neurons was adequate to induce localized type 17 immune response against Candida albicans, irrespective of any tissue damage or pathogen-related substances. The significance of pathogen recognition and the involvement of non-immune cells in initiating a functional immune response represent a growing area of research that links neuroimmune interactions with the host defense and inflammation. Candida albicans reactive T helper 17 cells found in both human blood and skin show a memory phenotype to ensure long-term immunity against commensal fungi and also to ensure efficient protection against reinfection with environmental fungi. In animals previously infected with or vaccinated against trichophyton, a delayed type hypersensitivity reaction can be observed, signifying the reactivation of pre-existing memory T cells specific to dermatophytes. And in human skin, T helper 17 cells specific to Candida albicans were found to express markers typically associated with tissue-resident memory T cells. Like T helper 17 cells, interleukin 17 producing gamma delta T cells are believed to develop long-lasting memory in scenarios involving interleukin 17 mediated skin disorders. They show heightened reactivity upon secondary challenges and display characteristics similar to memory cells. However, whether gamma delta T cell mediated recall responses are made by specific antigenic responsiveness or are more broadly reactive, like the case in trained immunity, this issue remains unexplored. The development of memory-like features in gamma delta T cells goes with the emerging concept of trained immunity, and whether gamma delta T cells activated in response to fungi establish a memory for an increased response to persistent or recurrent fungal exposure, similar to what has been observed in the gastrointestinal tract for protection against listeria, is a question that requires further investigation. And again, as my previous two videos, the main source of information in this video is this nice review, New Insights into Immunity to Skin Fungi Shape Our Understanding of Health and Disease, that was published in Parasite Immunology in February 2023. I hope that you have enjoyed this video, and I'm reminding you again to subscribe to the channel and activating the alarming bell, and thank you.